Assalamualaikum to our respected lecturer Sir Nazrul Hadi bin Ismail. In this video, my friends and I would like to talk about skeletal muscle glutamine. Firstly, I would like to introduce our group members that consists of Nurul Huda binti Mat Awi, Nur Izzati Ashikin binti Muhammad Hazian, and me, Siti Nur Amira binti Muhammad. So basically, what is glutamine? Glutamine is a L-alpha amino acid containing 5 carbons. It is the most abundant, free and non-essential amino acid in the body. Skeletal muscle constantly releases glutamine into the blood and is transported for use by other tissues through intertissue metabolic flux. Glutamine is needed to make other chemicals in the body such as other amino acids and glucose. Skeletal muscle is the largest organ in the human body, where 43% of the body's mass is made of skeletal muscle. It stores energy as proteins and amino acids that are broken down during catabolic conditions such as exhaustive exercises and starvation. The liver exhibits the highest capacity for glutamine production, and the skeletal muscle is the main tissue of glutamine synthesis, storage, and release. Glutamine is synthesized from the ammonia or ammonium ions with glutamate in an ATP-dependent reaction by the enzyme glutamine synthetase. Then, glutamine is reconverted to glutamate by a different enzyme, glutaminase, which releases the ammonium ions. This reaction is called glutamine hydrolysis. Next, we are going to talk about the rules for metabolism of skeletal muscle glutamine. Firstly, glutamine is the major transport form of ammonia. Glutamine is the principal source of ammonia in the kidney. The ammonia it produces enters the urine and decreases its acidity, and in this way, it plays an important role in the maintenance of acid-base balance. Glutamine also promotes the synthesis of heat shock or stress proteins which help protect body cells. It is also significant for intermediate metabolism, pH balance, and inter-organ nitrogen exchange via ammonia transfer between tissues. Next, glutamine metabolic pathway. Glutamine enters the cell through plasma membrane glutamine transporters such as SLC1A5, SLC38A1, and SLC38A, which can be used for the biosynthesis of hexasamine, nucleotides, and asparagine in the cytoplasm. Mitochondrial glutamate generated by the SLC25A18 and SLC25A22 transporters. Then, cytosolic glutamate takes part in the biosynthesis of glutathione. In glutaminolysis, glutamine will transported into the mitochondrial matrix by the SLC1A5 variant and eventually will be converted to glutamate by GLS, which is amido hydrolase enzyme, which catalyzes the conversion of glutamine to glutamate by releasing ammonium ions. Then, alpha-ketoglutarate is produced by the deamidation of glutamate catalyzed by glutamate dehydrogenase 1 or several aminotransferases including glutamic pyruvic transaminase 2 and glutamic oxaloacetic transaminase 2. Alpha-ketoglutarate exported from mitochondria through SLC25A11 to the cytosol 21 and participates in fatty acid biosynthesis and NADH generation. Then, mitochondrial alpha-ketoglutarate participate in the TCA cycle, supporting the oxidative phosphorylation pathway or the reductive carboxylation pathway. During oxidative phosphorylation pathway, after the synthesis of guanosine triphosphate and adenosine triphosphate, metabolites of glutamine participate in the generation of an electron donor, such as an ADH or FADH2. Reductive carboxylation of alpha-ketoglutarate will generate citrate is important for lipid synthesis under low oxygen condition, which is hypoxia. Alpha-ketoglutarate is an important cofactor for enzymes participating in epigenetic modification. It is a substrate for alpha-ketoglutarate dehydrogenase in oxidative reaction, which generates succinyl coenzyme A and isocitrate dehydrogenase 1 or isocitrate dehydrogenase 2, which then catalyze the reductive carboxylation reaction where alpha-ketoglutarate is being converted to isocitrate.